Blundell, coming from the University of Cambridge, and Jamie will be speaking about fitness consequences and mutation rates of Mosaic chromosomal alterations in clonal hematopoiesis.
Okay, thank you. Thanks, Ellie. Okay, can everyone hear me okay? Yeah? Okay, thanks for the chance to speak to you today. So yeah, my name's Jamie, I'm coming from Cambridge. I'm going to tell you a little bit about work that we've been doing in the last year or so on trying to understand the rates and fitness effects of mosaic chromosomal alterations. So these are things like gains, losses of large sections of chromosome that occur throughout your life, right? So they're somatic, they are not inherited. So generally speaking, my lab is interested in somatic evolution that occurs in normal tissue maintenance and in the run-up to developing cancer. So we're talking about pre-cancerous evolution here, and this kind of diagram has been seen already today. But the basic idea is that you inherit an ostensibly healthy pool of stem cells. This is a cartoon of, for example, blood stem cells on the left-hand side. And over the course of your life, in all of us, all the time, we're accumulating mutations, and in most of us, we are acquiring the first kind of cancer-driving mutations that drive clonal expansions. But some of those go on and acquire further mutations which drive further clonal expansions and eventually can drive, in this case, for example, the emergence of a leukemia. And what we're interested in doing is trying to understand this process, this dynamic process of going from kind of healthy all the way to cancerous in kind of all of its richness. And blood is a rather nice model for this because you have access to samples where you can actually watch this process unfold. So you do have, as I'll say in a minute, rather amazing serial samples. There's less spatial structure. I wouldn't claim that there's none. And there are already quite huge data sets generated from blood that give you an insight into this process. So today I'm going to talk very briefly about a framework that we developed a couple of years ago for estimating fitness effects from static variant allele frequency data. And then I'll move on and talk about how you can kind of adapt that framework to infer the mutation rates and fitness consequences of these mosaic chromosomal alterations. And then, and really that's kind of referring to the kind of first step in this process, right? We're talking about the acquisition and expansion of the first kind of cancer drivers in the tissue. And then I'll just flash up a couple of slides at the very end to kind of look at work that we're doing at the moment where you can kind of see the whole process unfold by looking at serial samples. Okay, so the basic idea behind the framework for inferring growth advantages for some of these mutations is the following. There's basically a lot of data out there from blood where you have really quite quantitative estimates of the clone sizes of somatic mutations from things like whole exome sequencing data. Okay, so these are mostly kind of large data sets that have been sequenced to maybe 100 or a few hundred X. And what you can do from that is you can call quite confidently the somatic mutations that have arisen in this case, blood. Okay, and so this is what I'm plotting on the kind of, the y-axis here is the variant allele frequency, that's just a measure of how big the clone is, and on the x-axis there is just the position in this particular gene. And what you see from this is, by the way, cancer-free individuals, 50,000 of them. But what you see from this is that there's a huge diversity of clone sizes that you see. Most clones are actually very, very small, some get big. And the basic idea is that there is information in that about the forces of mutation and selection and genetic drift that are occurring inside the tissue. And so to try and understand those, what we do is we link this data with kind of simple models of stem cell dynamics which have those three components, right? You have the acquisition of mutations, the stem cells undergo symmetric cell fate decisions, and so that drives genetic drift and extinctions and things like that, but you can also acquire mutations that will make that process imbalanced and can drive clonal expansions. Those are the kind of important features. Now, the important kind of take-home from the framework is that the variant allele frequency distribution that you see in a tissue, so this is plotting basically the number of variants you see at a particular position across this large cohort of people as a function of variant allele frequency. So it's the clone size distribution for a particular variant. And the point here is that contains information about are these forces. So, for example, the distribution you expect under this simple model, at low frequencies, low variant allele frequencies, the number of clones increases rapidly, so it increases as kind of 1 over the frequency. But the amplitude of that tells you about the product of the number of stem cells in the tissue and the mutation rate to that mutation. And so because the mutation rate to that mutation is well characterized in blood, you can then back out the population size of stem cells that maintain your blood. And in this case, you basically infer there to be about 100,000 or so stem cells. The other important feature is that the number of variants you see as a function of frequency begins to fall off as you go higher and higher in frequency. And the position at where it begins to fall off tells you something about the growth advantage conferred by that mutation. Okay, so you can also read this off, and in this case, this particular mutation we inferred to have a fitness advantage of about 15% per year. So the key take home from this is that you can begin to put numbers on some of these key parameters that drive the evolution of somatic evolution in these tissues. So we think there's about 100,000 stem cells that maintain the blood, and this has been validated through other studies. And the other kind of big point is that it looks like in blood at least, the evolutionary dynamics is selection dominated, not drift dominated. Okay, so the selection effects of these drivers are typically 10 to 20% per year. In a population size of 100,000 or so stem cells, it's strongly selection driven. Okay. So why did we begin thinking about MCAs? The point of MCAs was that we did a piece of work where we asked the question, how many clonal expansions are there in blood anywhere in the genome? And the way we did this is by looking at synonymous mutations, because they act as kind of neutral barcodes, and they give you an unbiased estimate for the total number of clonal expansions in the blood, independent of whether you've seen them or not. And the basic take-home from this piece of work was that we were estimating that most clonal expansions in blood are not occurring, are not being driven by SNVs in cancer-associated genes. And this actually might relate to something that Roberta mentioned in the second talk, that we think there's likely a lot of selection outside of the canonical cancer-associated genes. And this work, that number actually has been validated in recent studies using single-cell phylogenies. So that, coupled with the fact that Mosaic chromosomal alterations are common in blood, and there's work from Ellie's group and others which has really beautifully shown that, the question then that we asked is, to what extent can this missing selection be explained by these Mosaic chromosomal alterations, and what are the mutation rates and fitness effects of them? Okay, so what we did is we turned to this amazing data set generated by Lowe and colleagues at Harvard, and this is where they have called somatic chromosomal alterations from the biobank, from all participants in the biobank, from SNP array data, that they have, because they have phase, they have good haplotype information, they have really exquisite ability to call quite low cell fraction events. And so this is all of the data plotted here. So this is, gains in red, losses in blue, copy neutral, loss of heterozygosities in orange. And what you see again is this telltale density which increases very rapidly as you go down in frequency. But because this data is so quantitative, you can really begin to pick apart things about the mutation rate and the fitness effects. So again, this is the density of chromosome 14q gain, for example, here. This is just plotted on a linear scale, and the prediction is that should be a 1 over f divergence. The high frequencies should decay exponentially, and you can really quantitatively check that. So if you plot it on a log scale on the y, you basically see that exponential decay. If you plot it on a log-log scale, then the 1 over f divergence should be a flat line. And you again see that it is rather nicely flat. But the important point there is that the position of the amplitude of that flat line is telling you something about the product of the number of stem cells and the mutation rates. But the mutation rates of MCAs are far less well characterized. But because we now know the number of stem cells, we can back out the mutation rate for that event. So if you renormalize it, you can see that the gain of chromosome 14q gain, we estimate to be occurring at about 1 in 10 to the 9 per cell per year. And when it occurs, it expands about 14% per year. So the point here is that you can begin to disentangle the role of selection versus mutation in this, and I'll just show a few examples of these, right? So for example, the top here we have 9q loss. 9q loss is quite rare. We infer it to occur quite infrequently, 3 times 10 to the negative 10 per cell per year. But when it does occur, it seemingly expands rather rapidly. So if you look at the distribution, the fall off goes right out to quite high frequencies, and we estimate that That means it has a fitness advantage of about 15%. But the opposite is true for the gain of 21q, for example. So that's quite common. It occurs at kind of 2 or 3 times 10 to the negative 9. But they're almost exclusively confined to low variant allele frequency or low cell fraction, meaning that they don't expand very rapidly. Right, so you can do this across all of them, and you can build essentially a league table of the ones that you think are the most fit. And what you can do then is build up a kind of quantitative picture of the distribution of fitness effects broken down by mutation rate for the gains, for the losses, and for the copy-neutral loss of heterozygosities binned by chromosome arm here. So what you basically see is that it seems as though gains and losses occur at a lower rate, but confer greater growth advantages to the cells compared to copy-neutral loss of heterozygosity events, which occur much more frequently, but confer a much more modest selective advantage to the cells. Why may or that may not be important is that it looks as though there is at least a weak correlation between the growth advantage conferred by the change and the subsequent risk of developing a blood cancer. And that kind of stands to reason. If it grows more rapidly, it has a higher probability of acquiring further genetic changes that may drive a cancer. But the place where I think this may actually have its be most useful is that it serves as a kind of simple null model for identifying interesting behavior, right? And so what you can do with this model is it can predict what the age dependence of these events should be. So it basically says, look, if they're growing over time, then there should be more old people who have these events than young people, and it predicts how that curve should go. And if you group all of the gains together, all the losses together, all the copy neutrals together, it kind of fits okay. It doesn't fit great for the copy neutrals. It fits okay for the others. But inside that are examples where it fits exquisitely and examples where it completely fails. And so just highlighting a few of these, right? 10q loss, the model gets completely wrong, right? So the model predicts that it should be quite strongly age dependent, and that age dependence should be different in men versus women. But if you look at the actual data, which is the data points there, there's almost no age dependence at all. And so the model is just failing spectacularly here. But that's interesting because it says, okay, there must be something else going on, or very likely something else going on. And in the original study, this was actually highlighted because it was one of these changes where it was strongly associated with an inherited variant that was acting in cis in this case. So there are other examples that we think you can highlight which are going to point to kind of new or interesting biology. So this is an example where 20q copy neutral. In women, it looks like it fits rather nicely. In men, the fit's horrible. There's no age dependence. There presumably is something interesting and new going on there. Okay, and then the last thing I want to say is that, so what I've shown you so far is that you can begin to leverage some of these large data sets to learn something about the growth advantages and the mutation rates to these early mutations, the mutations that accrue in your blood stem cells and begin to expand. And what we're trying to do is kind of bring that together and understand that full dynamic process that takes you from a healthy piece of tissue to a completely cancerous one. And what we've done is we've teamed up this remarkable study led by Usha Menon in London that has recruited 200,000 postmenopausal women and sampled them. They've given blood every year for 10 years. And because it's such a large study, many of them developed all sorts of incidental cancers. And so we then coupled that by developing kind of error-corrected sequencing strategy where you can detect very low variant allele frequency SNVs and MCAs. These are the samples here. And I'll just flash up a slide. This is not, we're writing this up at the moment. But you see this amazing evolutionary dynamics that occurs in the run-up to the cancer. Like really rich clonal interference that is occurring decades before the diagnosis, or at least nearly a decade or so. If you're interested in that, I can show you more after the talk. Okay, so right. So what I've tried to tell you about today is a framework that we've developed, which we think can begin to estimate the kind of key evolutionary parameters that drive the somatic evolution in tissue, in this case blood. You can estimate the fitness effects of variants at single nucleotide resolution, and you can begin to infer what's the population size of stem cells that maintains the blood, and that kind of is important because it informs the strength of drift and things like this. And most of these seem to validate in time series data and single cell phylogenies from other people's work. I've then gone on and told you about mutation rates and fitness effect of MCAs. The key take home here is that the fitness effects of the MCAs and the SNVs looks kind of similar, right? They confer growth advantages in the kind of 10 to 20% per year range, but the MCAs occur much less frequently than the SNVs. So the mutation rates of the MCAs are about tenfold lower than the SNVs. So actually, MCAs, we think, are not explaining much of this missing selection, and there's probably all sorts of other drivers elsewhere in the genome that we're just not seeing at the moment. It also is kind of intentionally simple. It's obviously not going to contain all the richness of the biology, and it has many, many limitations. But by the examples, the counterexamples which don't work, should highlight places where you can look for more interesting biology, and that includes extrinsic factors and things like inherited variants which shape the mutation rate and fitness effect. And then lastly, I just showed some longitudinal data that we're doing, which shows that the evolutionary dynamics is different in people who develop cancers. Okay, and I will end there and just highlight Caroline Watson, who's on the left-hand side here, who's driven a lot of that work, and Gladys Poon, who's nearest to the camera, who has done a lot of amazing work on the missing selection. And just to highlight that we have just established an early cancer institute in Cambridge, which is a huge priority for the UK and for CRUK generally. And we hope to be recruiting about 2 or 3 faculty over the next few years. So if you're interested, then please get in touch. Thank you.
Thank you very much, Jamie. So I would like to start with a question. That's all right. So excellent talk. Thank you so much. So you alluded to it in your discussion, but both Pure Low's data set as well as our data set has shown that about 18% of Mosaic chromosomal alterations tend to co-occur with underlying predisposition variants, and we've estimated a conservative kind of 23% of MCAs to overlap acquired somatic mutations. Yeah. And kind of in your last example, you've showed a case where there was a JAK2 mutation and occurred a 9p-LOH, which overlaps the JAK2 locus. So how do you incorporate this information into your fitness estimates when we know, for kind of the benefit of everyone in the room, that many of those MCAs specifically co-occur with pre-existing germline or somatic alleles? So that's my first question.
Yeah, so no, that is a really important point. So we don't have access to the data of the somatic variants from the exome. Well, in principle we do, but we haven't looked at that yet. But that's so the interaction between somatically acquired SNVs and the MCAs, as you've already found, clearly is there, and it will affect some of these inferences, but we haven't looked at that in detail in the biobank data. You do see that effect from the germline, right? So I think one of the big caveats to what I've presented is that the copy-neutral ones, the copy-neutral fitness effects are weird, right? Because you're not changing the dose. So that must be strongly influenced, because the genetic change is going to be influenced then only by the inherited variants that you have on that bit of the chromosome. And so clearly there must be some dependence on the inherited variants that person has. And so, whether the model is appropriate for the copy neutrals is a little more questionable there.
Because I struggled a little bit with the notion that amplifications and deletions have stronger fitness effects than LOH, because we know that each of them target different alleles in their own way. And perhaps, given the fact that we've established a good understanding of a subset of these loci that tend to have a very strong concurrence with pre-existing allele, I wondered even whether in the absence of the mutation data, you can look specifically at that loci 4q, 9p, 17, and 7, and see if you do see differential effects in fitness there compared to the kind of genome-wide distribution of MCAs.
We have looked a little bit of that, and that is in the supplement of the preprint. So you can begin to do a bit of that, but the results are not the most very conclusive there.
Okay, So any questions? We have a question back. Please identify.
Very interesting work.
Yourself.
I'm Hossein Kiabanian. I'm from Rutgers. Very interesting work over the years. Following up with that question, basically, are there any, if you look at the tumor data, do you see recurrences of these amplifications and deletions, or general copy number changes? And have you looked at the targets, genes that overlap with these regions, the same as JAK2 situation, for example, on chromosome 19?
Yes, we have a bit. So that was also kind of covered a little bit in the original paper that called the MCAs. In some cases, you can clearly identify it just by eye, right? I mean, like chromosome 14, you have the loss of DNMT3A, or loss of sections that overlap DNMT3A, and you have this like beautiful kind of like funnel-like thing for that. In most cases, I don't think you can actually pinpoint it to a particular locus. What was your first? Sorry, I forgot the first bit of your question. Oh, in cancers, that's right. So most of these people are so all these people are kind of healthy when they gave the sample. Most of them do not develop blood cancers or even blood cancers. Some of them do, and that's how I got that kind of whether it informs your risk. But we have not. It's actually we have not looked into getting the actual data from those blood cancers. I'm not sure we would be able to do it actually. Yes, so a lot of them are well known in hematological cancers, yes. Yeah.
Right, thank you so much, Jamie.
Great, thank you.
So with this talk, I would like to thank all 4 speakers from this morning's session for giving excellent presentations of their recent work and kicking off the day very nicely and very dynamically. So we now have a 10-minute break,